Thank you, Chris. Welcome to Café Scientifique, Silicon Valley. As you know, we meet on third Thursday of every month, January through October, here, graciously hosted by HanaHaus. I'm Byron Brown, proud husband of Kay Brown, who organizes these talks. Not to worry, she's in good health and voice, but wanted me to have the privilege of introducing tonight's speaker. He is Dr. Ronald Levy, professor and chief of the Division of Oncology of the Stanford School of Medicine. His interest in the immune system began early in his medical career and deepened during his work at the Weizmann Institute and the National Institutes of Health. Throughout Dr. Levy's career, he has remained convinced that the body's own defenses could be used to fight cancer, even though cancer cells often fail to trigger internal defense mechanisms. In the words of the American Association for Cancer Research, quote, "He has been called the antibody hero with good reason. After decades-long quests to find a way to use the body's immune system to develop antibodies against invading tumor cells, Dr. Ronald Levy helped develop the first FDA-approved antibody treatment for lymphoma. Rituximab entered the market in 1997 and proved successful in half the patients to whom it was given, resulting in tumor regression and very few side effects. Rituximab is now used for every lymphoma patient, either as a standard treatment or in combination with other therapies. Dr. Levy has received at least 15 major awards for his work, but I rather believe his best reward is in the lives he has saved and extended. And I'm very grateful to count mine among them. 6 years ago, I developed large abdominal tumors which were diagnosed as stage 4 non-Hodgkin's lymphoma involving multiple organs. After just 2 chemo sessions with rituximab, a positron emission tomograph could not detect a single tumor. I've enjoyed a very healthy 6 years ever since. So it is with great heartfelt gratitude, it is my honor and pleasure that I now present Dr. Ron Levy.
Thank you very much for that nice introduction. I hope everyone can hear me. We're going to have a question and answer, but we're going to save it for the end because I have some stuff I want to tell you and get through what I want to show. So to begin with, I'd like to know who I'm talking to. So how many of you are scientists in the audience? Raise your hand. Uh-oh. Okay, a lot of ringers. How many are doctors? How many are in the biotech industry? How many are stock analysts? How many have heard of Rituxan? How many have received the drug Rituxan? Okay, so I think I don't need to tell you anything that you don't already know. You already know probably the whole story. Okay, so if I accomplish nothing else tonight, I think by the end of the talk you'll understand why these 2 individuals won the Nobel Prize for Medicine this year. They received their prize in Stockholm. Jim Allison and Tasuku Honjo, one from the Berkeley Bay Area who now lives in Texas, who sings in a rock band called the Checkpoints, and the other one, a famous immunological scientist from Japan. So to begin with, I want everybody to understand what you need to know, all you need to know about the immune system of the body. So in your blood, there are billions of what we call B cells and T cells, each one of which has a receptor on its surface that recognizes foreign things that come into your body and rejects them, like viruses and bacteria and foreign objects. And there are billions of each of them in the blood, circulating around all the time. As our colleague Irv Weissman likes to say, the real purpose of the heart is to circulate these cells of the immune system around the body, to be on the patrolling for infections. So this was a system that inherited through evolution to teach our body how to fight off foreign invaders. But cancer is an invader from inside your body. It's an invader, one of your own cells in your body goes haywire, has mutations in their DNA, and then goes into wild replication and makes more copies of itself and gets you in trouble by winding up in the wrong places in your body and interfering with the way your body works. And so when I began my laboratory at Stanford over 40 years ago, I wanted to use the immune system to try to fight the invaders from inside as well as the invaders from outside. And we knew a lot about antibodies, and we wanted to try to use antibodies to treat cancer. And most people those days thought this was a terrible idea. They thought it could never be a drug. An antibody is a big protein, you can't eat it, you have to inject it, and has all these problems. I said there are billions of those cells that make those antibodies in your body, and everyone makes a different antibody. Every cell makes a different antibody. So when you have a mixture of all those products from the blood of an animal, let's say, it has billions of different antibodies. And so every time you get it from an animal, it would be different, a different mixture. Never be the same. It's a big molecule, it's a protein. It doesn't get inside cells, even if you inject it in the body. Too big to go inside the cells. It was hard to make. And originally these were foreign proteins, so they would be seen as foreign invaders in their own right, coming from the outside. And maybe they were a little bit too specific. So each one of these problems got solved. And I'm going to tell you about the main solution to the problem at the top here. But every one of these problems eventually got solved. And now antibodies, we have a lot of drugs that are made from antibodies. And I'm going to tell you the story of the first one that we originally made. But the solution to the problem of they're always different and they always come from one original cell was figured out in England by two scientists who figured out how to capture the cells from the body of an animal that had responded to a foreign substance. And from the spleen of that animal, where those immune cells are, they were able to clone those cells one at a time. And they cloned them by fusing them together with a cancer cell. Cancer cells live forever. They never die. As long as you give them enough food and keep them happy, they live forever. They have immortality, ironically enough. But the property of immortality from the cancer cell and the property of making one and only one antibody from the immune cell were fused together, and then we had what we call hybrid cells, hybrids between two cells, that live forever and made only one antibody. And so we can make billions of these one at a time, and we can have them forever. We can make as many of them as we want, many copies as we want, because the cells have immortality now, they live forever. And so then it became a problem of what are we going to make the antibodies to recognize? What do we want them to recognize in cancer cells? And so we wanted to find targets in cancer cells that are different from normal cells. So if we have a cancer of the immune system, cancer of those same cells, it will live forever, and it makes its own antibody, its own unique antibody, and that will provide a target which is only on the cancer cells and not on normal cells. And so that seems like a perfect target to go after. So we made antibodies using the technology that I showed you from the two scientists in England. We made monoclonal antibodies that recognize the target that's only on the tumor cells of leukemia and lymphoma patients. And we made a bunch of these antibodies, and we eventually wanted to use them as a drug. So my teacher at the time, Henry Kaplan, who some of you know, I know that there are people in the audience who remember Henry Kaplan. He was my teacher. And he said to me, when he heard what I was going to do, he said, "It's very important that the experiment work the first time." And I think he was talking about one of his own experiments, not mine. But the experiment that worked the first time is shown here. His name was Philip Carr. He was 67 years old at the time. He had non-Hodgkin's lymphoma, and we had his cells from his tumor, and it had that target that I described, and we made a monoclonal antibody using the technology I described, and we made lots of it, and we had it in the freezer, and we waited. We waited about 2 years, and he was getting the regular treatments that we always use, the chemotherapy and the other treatments, radiation therapies. And eventually the treatment stopped working, and then we took the antibody out of the freezer and we treated Philip Carr with this antibody. This had never been done before, and now that looking back on that, I realized that we broke a lot of laws that we were ignorant of at the time. We were stupid enough to go ahead, but it worked the first time. And so I know one of our fellows in the audience here will recognize what he's looking at here. This is an X-ray of Philip Carr's body, and you see the spine here and the pelvis here, and you see these things here that are extra. These are lymph nodes that are full of lymphoma throughout his body and in this area here. And it's a special X-ray that we don't do anymore, and we can go into the reasons why we don't do this X-ray anymore, but it shows what happened before and after we gave him that antibody, June of 1981, September of 1981. Now those lymph nodes have gotten smaller, and they've gotten brighter because the dye can be brought together and seen better. And so this is almost what normal lymph nodes look like, and this is what lymphoma lymph nodes look like. And just by giving him that antibody that we had made, he went into what we call a remission of his disease, and that remission lasted for over 30 years. Mr. Carr died 2 or 3 years ago in his mid-90s, and he never had lymphoma again, just from giving him that antibody we had made. So we published this result, and that gave rise to what we call today the antibody biotech industry. So we started a company based on this idea, a company called IDEC Pharmaceuticals. And IDEC made, for about 50 more patients, custom-made for each patient, an antibody that we used to treat those patients. And we had many good successes with those antibodies. But after about a couple of years, the company decided that it could never make money doing this. Making a drug for every person, unique for every person, even if the target was perfect, and even if it worked perfectly every time, which it didn't work perfectly every time, but they thought they could never make a profit doing that because it was so difficult and expensive. Looking back on that now, many years later, and revisiting that idea, I think it's time to open that idea again. Instead, they wanted to make an antibody that would be good for everyone, ready to go, off the shelf, and really available. And they made an antibody against the target that we call CD20. And I told them that was a terrible idea. I told them that we would lose all of the special features of the target we were going after. And this target is on normal cells in the body of the same type, so-called B cells, B lymphocytes. But they did it anyway, and we gave it to a monkey, and nothing bad happened to the monkey. So we did a clinical trial with this antibody. It now is called Rituxan, and I think many of you know about this or have received this drug. Rituxan was a huge success. About half the patients who got the drug went into remission. The remissions lasted a year or two at a time, and there were very few side effects. Now, clinical trials are usually approved by the FDA when they're randomized. Half the people get it and half the people get something else, and you see which is better, and then if you can prove yours is better, then they get approval. Rituxan was approved on what we call a phase 2 clinical trial. We just gave it to a bunch of people, they had a response, and they didn't have side effects, and the FDA approved the drug. I think that'll never happen again. It was a lucky, at the time the FDA didn't really know anything about antibodies, and they're so impressed with the lack of side effects. So let me tell you an anecdote. So Wendy Harpham was a patient of ours. She was an internal medicine physician when we first met her. And her story is summarized here. She was 36 years old when we met her. She had a diagnosis of a certain kind of lymphoma. She failed chemotherapy and radiation therapy. And she was one of the first patients on the clinical trial that we did with the antibody that became known as Rituxan, a phase 1 trial. And she had a partial response of all her tumors in her body. And then it came back a year later, And then she got it again, Rituxan again, and she had a complete regression of her tumors throughout her body. And then in 1995, a year later, she got it again. She got chemotherapy, and then she got Rituxan again in 1997, and again in 1998, and in 2005. And in 2007, she got it yet another time, and she went into remission, and she's never relapsed her disease ever since. So I was talking to a patient in the clinic today who asked me, if I get Rituxan now, can I get it again in the future? And I said, what are you doing at 5 o'clock tonight? Come to my lecture. And she's sitting here in one of the back rows. So that's my anecdote that I wanted to let you know about. So here's Wendy Harpham today, 26 years later. She's a grandparent, and she made me promise to show people these slides, these pictures, especially if they would encourage other people to join the clinical trials, students to embark on careers in cancer translational research. And I'm fulfilling my promise to her by showing you these slides today. So as I said, Charles Schultz knew the answer back then. Aren't antibodies wholesome, natural, and without side effects? And that was the thing that really impressed the FDA at the time, the lack of side effects. In comparison to all the other treatments that we have for cancer, these antibodies are very safe to give. And that was completely not predicted. As I told the company at the time, it was a terrible idea, and I'm admitting that now, many years later, it was actually a good idea and I didn't recognize it. But the difference between a treatment that is off the shelf and ready to go, that now it's a $5 billion a year drug, used throughout the world for every patient with lymphoma, compared to a custom-made boutique product, one patient at a time, that never saw the light of day commercially. So that lesson is really important. If you have a new treatment or a new thing, it has to be practical, it has to be cost-effective, it has to be made available to really change the world. So that was a big surprise that it worked at all, that it was safe, and now it's a big drug. But the next big surprise, one day a patient came to me who had arthritis, rheumatoid arthritis, and he also had lymphoma. And so we gave him Rituxan for his lymphoma. So he came back a couple weeks later and he said, I don't know about my lymphoma, but my joints feel better. And that was the beginning of the realization that this drug is not only good for lymphoma, but it's also good for autoimmune diseases like rheumatoid arthritis. And here is the list, a short list, of all the diseases that are being treated today by Rituxan. And many of them are caused by immune system and antibodies. Many of them were not thought to be caused by antibodies, but are like multiple sclerosis or rheumatoid arthritis. But they do respond to this treatment. And now, if I got my facts straight, the worldwide sales of Rituxan, is more for these kinds of diseases than it is for lymphoma. So that was another big surprise. There are many surprises in this story. And for each of these stories that became a success, there's a story to tell about how it could have failed, it could have missed the mark, it could have not had the lucky accidents that I talk about here. One of the lucky accidents that happened with Rituximab was the company that originally I referred to, that originally was founded on that boutique idea. That company was having trouble raising money and was almost going bankrupt. And I went to a conference at Stanford where industry and academia were kind of meeting each other, and I sat at lunch randomly next to this guy, and he said, what do you do? And I said, tell him what I did, and I said, what do you do? And he said, I work for Genentech, I'm a business developer. And he said, oh yeah, we've heard that story that you're working on. That's IDEC, and we decided not to invest in that. And I said, I think you should take another look. I think this is working in patients. And he said, okay, I'll bring my bosses by and they'll talk to you. And about a week or two later, they came by and talked to me, and I showed them the results. And a month or two later, there was a deal struck between Genentech and IDEC that at the time saved both companies. And so now they're fighting over who gets all the royalties of this successful drug. But they were both successful because of it. So there were a lot of lucky accidents along the way. There are more lucky accidents, but I want to move on from the Rituxan story to getting to the Nobel Prize and those two winners this year and explain to you what they did to win the Nobel Prize. So I've been talking about antibodies, and they come from these cells called B cells in the blood. Now let's talk about T cells. T cells are the other arm of the immune system. They also have a receptor, like an antibody, but that receptor stays on the surface, doesn't come off the cell. And that receptor recognizes something different. It recognizes cancer cells or abnormal cells, and I can go into detail later in questions how it does that. But then it kills, or it does things bad to the target cells that it sees. So that's also a potential treatment for cancer, because if this were a cancer cell, and this were a T cell that has the right receptor, that recognizes it, it could kill that cancer cell. All we need is billions of these T cells that can do that job. So this guy, friend of ours, named Zelig Eshar, working in Israel, got the idea that he would take the receptor from a T cell and the receptor from an antibody and put them together genetically. This is the antibody part, and this is the T cell receptor part, coming from the T cell receptor. Make one receptor that knows how to recognize things that the antibody can recognize, but signal the T cell to kill the way a T cell receptor would do. And this is called a chimeric antigen receptor. It's chimeric because it has two parts, one coming from an antibody, one from a T cell. And he put that gene into T cells and showed that those T cells can now kill whatever the antibody could recognize. So we have a T cell that's been engineered outside the body and can be used to kill the cancer cells that the antibody can see. And so he teamed up with this guy, Steve Rosenberg. He works at the National Cancer Institute, and the two of them created a product custom-made from the blood of a patient. Remember, I'm saying custom-made again here. Custom-made from the blood of a patient, took the T cells out of the patient, engineered them with that receptor, that chimeric antigen receptor. So took the blood out of a patient, got the T cells from the blood, put the gene into those T cells, made a whole bunch of them, billions of them, and then gave them back to that same person in a circle, coming from him, engineered outside the body, going back to him. Very expensive, very laborious, very hard to do. And the question is it worth it? So here's the proof that it works. This is a young girl. Now she's a teenager, I believe. Her name is Emily Whitehead. She had leukemia and was dying of leukemia, and they did that custom job for her in the University of Pennsylvania. And they treated her with her own T cells that were engineered outside her body with that receptor that I described, gave them back to her, and her leukemia went away and she was cured. So here's a picture of her 2 years later when she was on her way to visit the president of the White House. Not the current president, President Obama. And now, as I said, she's a teenager and has a she is literally the poster child for this whole idea. So now chimeric antigen receptor T cells have made it big. They really work, and they're really hard to make, and they're really expensive. So the diseases that we have chimeric antigen receptor T cells for are acute lymphocytic leukemia, where the results are dramatic, B-cell lymphomas, where I have here worthwhile. I think I can replace that now and say more than worthwhile, really work very, very well. And they've been approved by the FDA on a customized basis, one patient at a time, to make a product for the therapy of their cancer, their leukemia, and their lymphoma out of their own immune cells from their own body. So the target here is a target called CD19, different from the target of Rituximab, but it could be the target of Rituximab. It's another target that's on these cancer cells, leukemia cells. There might be other targets on other kinds of cancer that could be made into chimeric antigen receptors, and many people are working all over the world on this whole idea. We just came back from China, where we were told that they're the leaders now in this whole technology, and they're going to beat us to the punch in doing. We'll see about that. One of the problems with this idea is that, number one, it's custom made and very expensive. You have to wait several weeks to a month, or even longer, to get these cells made for you, and you have to be able to wait. They're not off the shelf. And there are side effects. When those T cells go in the body, they cause a lot of fever and a lot of blood pressure problems, and sometimes people even have to go to the intensive care unit to take care of them to get over this. So it's a treatment that has to be done in the hospital, and it's very expensive just to take care of the people. And there's also problems in the brain that come from these treatments. And it's very, as I've been emphasizing, expensive and impractical. But maybe the problem will be solved to take T cells from somebody else and engineer them and give them to whoever needs them. Then we could have them on the shelf and ready to go, and it would be a lot cheaper. And there's many people working on solving that problem now. So this whole subject, you read about it in the newspapers, CAR T cells curing leukemia, and it is a big success, but we have a lot ways to go to make it even better. So I want to shift now. Everything I've been talking about now is targeting the cancer cell. Either the antibodies go there and latch on and destroy the cancer cell, or the CAR T cells go there and kill based on the killing power of the T cells. So, and all our drugs that we have in the clinic are targeting the Achilles heel of the cancer cell. The big paradigm shift now, and what led to the Nobel Prize, was the realization that we can make antibodies that can target the immune system and get the immune system re-educated to go after and do the job and kill the cancer cells. That realization was made by this guy, Jim Allison, a number of years ago when he was working at Berkeley, University of California, Berkeley. Subsequently, he moved to New York, to Sloan Kettering. Now he's back to his home state in Texas. And I told you he has a rock band. He's a singer and a harmonica player. He kind of looks like a rock band singer. But he's really an amazing scientist, and he made the discovery that you can take, that the immune cells of the body are being, this picture is a little too complicated, but basically the immune system of the body has a set of stimuli that make it go, but it has a set of brakes that turn it off. And this target is the brakes on the immune system of the body. And so he discovered that, and then he made an antibody, a monoclonal antibody, that take the brakes off the immune system, and then the T cells can go wild when you take the brakes off. Now, if they're the right T cells and they go wild, they can attack the cancer cells. If they're the wrong T cells and go wild, they can attack other parts of your body. And that's the downside, the risks and the benefits of doing this. About the time he did that, very soon, maybe almost the same time, the other guy I mentioned from Japan cloned a gene that he called PD-1. PD-1 is another property of the T cell, the immune T cell. And this PD-1 receives a signal from the cancer cell, and that signal says, don't kill me. So the immune cell maybe can see the cancer cell, but the cancer cell is giving a signal saying, I know you can see me, but don't kill me. And so a number of other people, including the guy from Japan, made monoclonal antibodies against PD-1, or the receptor for PD-1, called PD-L1. And that has changed the world. That is an antibody off the shelf, good for everyone with cancer, because everyone has an immune system. Maybe everybody has a different kind of cancer, but everyone has an immune system. And that treatment against PD-1 or PD-1 ligand has really changed the world. Here's an example. This is how we track clinical trials. These are patients with metastatic melanoma, a malignant lethal skin cancer, and it shows every patient is one line here, and it shows what happened to all the patients that were treated with these antibodies against PD-1 and the original target, CTLA-4. And see how many patients shrunk their tumors here just by talking to the immune system of the body, taking the brakes off and letting it do its job, letting it do the job we want it to do. Most of the patients had shrinkage of their tumors. A few did not, but most of them did. And here's an example of abnormal spread of the cancer under the armpits of the patient. going away in 12 weeks after getting these treatments. This is a disease for which up to this point in time we had no effective treatment of any kind. Chemotherapy didn't work, all kinds of other things didn't work, and finally we have a treatment that everybody with melanoma is now getting this treatment, everybody throughout the world who can afford this drug. Even better, even more important, lung cancer. Lung cancer is much more a public health problem than melanoma. And here's a clinical trial, early clinical trial in lung cancer patients. And here it's a different kind of a graph. Every patient is a different line here. If the tumors are growing, the line is going up. If the tumors are shrinking, the line is going down. And here are some examples showing an abnormal tumor in the lung of a patient going away. So this is anti-PD-1 antibodies given to lung cancer patients. Look how many of them had their tumor shrink just by talking to the immune system. And the green lines are patients who never had any other treatment of any kind for their lung cancer before. This has really changed the world. This has changed the way lung cancer is treated throughout the world now. And it's even used in people like the green ones here who've never had any other treatments for their lung cancer. So you can imagine why these guys won the Nobel Prize. Now here's a list of a short list of the diseases that, the cancers, all the different kinds of cancers that, for which this single antibody, off the shelf, ready to go, can work. From lymphomas of Hodgkin's disease to all kinds of other very dangerous cancers who are now being treated. And the ones with stars next to their name are the ones that have been approved by the FDA for these kinds of treatments. So I think you can appreciate how the whole idea of using the immune system to treat cancer has progressed from targeting the immune system against targets we know to talking to the immune system and taking the brakes off and let it find its own targets, whatever it wants to see. Incredible progress just in the last few years. There are a lot of questions about this that we can make it better maybe. Not everybody responds. Some patients respond and some do not. Some kind of tumors respond and some do not. Lymphoma, but not myeloma. Stomach cancer, but not colon cancer. So we want to make this all work better. And we want to figure out the reasons why it doesn't always work. And it has side effects also. Taking the brakes off the immune system, as I implied, if you take the brakes off the wrong part of the immune system, it can attack other parts of your body and make autoimmune diseases. So we've created business for the rheumatologists, the dermatologists, the liver experts, the colon experts, the lung experts. They're all got autoimmune diseases now that we're creating by giving these take the brakes off kind of treatments with these PD-1 antibodies. So we have to reduce the side effects of all of this. Sometimes the side effects can really be serious. And so that's a lot of what's going on. So that brings me to, someone once told me that you're only as good as your next idea. And so Rituxan is the story of the past. It's a big success. What's my next idea? What are we doing now to take advantage of everything that's been learned and try to address some of these problems of the side effects and making it work better and using the immune system with treatments that are off the shelf and not custom made for each person. So here's what we're doing now. We're trying to treat cancer in one place in the body with some of these antibodies that I've been talking about that take the brakes off the immune system, but take the brakes off only in the area of the immune cells that are in and around the cancer cells, and not all the immune cells in the body. Those are the ones we really want to make go. And we don't want to make the other ones go because they cause the side effects. So if we can inject those antibodies directly into the tumor where the immune cells that are there are trying to kill the cancer, but the cancer is trying to say, don't kill me, take the brakes off right then and there and let those immune cells go around the body, patrolling around, looking for cancer on the other side of the body to show that it can do the job all over the body. So that we call this vaccination against cancer in situ, right in the place where the cancer is. So that's what we're doing now. This is a mouse, as you can imagine, not a person. And we've been doing experiments in the lab now to try to find the very best things to be injecting into the tumor microenvironment so we can trigger that immune response and get it going, not have to do anything outside the body, not have to know the target in advance, and a treatment which is off the shelf. As we say, all it has to do is work. So I can tell you it really works well in the mice. We've found this combination that I'm put on the slide here, this combination of things that we can inject into the cancer and get not only the place we inject to go away, but get cancers all over the body to go away in the mice. So, having done it in the mice, let me show you an example of a result. This is a kind of mouse that gets breast cancer. A little hard for you to see, but the arrows here are pointing to all. The mouse has 10 mammary glands. I didn't know this before, but 10 mammary glands every mouse, 5 pairs. And in this genetically rigged mouse, every mammary gland gets a breast cancer by about 3 months of age. And these are independent, arising in each of the mammary glands of the mouse, very malignant. They spread to the lungs, and eventually the mice die. Every single mouse in this genetically rigged strain gets breast cancer. So what Idit Segiv-Barfi, our brilliant postdoc from Israel, did was she actually watched the mice and watched to see the very first one breast cancer they got. She injected it with this take the brakes off signals, and then looked to see what happened in the mouse, in the rest of the mammary glands that would normally be getting cancer. And so she was able to cure and prevent the breast cancers that were about to happen in these mice by injecting the take the brakes off signals right into the first one they got. And so we're so excited about this result in these mice that we're now checking it out in people. And so what are we doing now at Stanford? We have clinical trials that we're doing in patients with lymphoma and with other kinds of cancers as well, where we're doing exactly what we did in the mice. We're taking those very same take the brakes off signals, we're giving them to one place in the body where the cancer is, shown here, and we're tracking the other places in the body for whether they're going to respond or not. And I can tell you that people are not mice. It doesn't work quite as well in people as it does in mice, but it's working well enough to really whet our appetite to really expand and do this more. And so we do this with a whole bunch of people who are listed here. I think maybe some of them are even in the audience. A big team of people, doctors, scientists, nurses, radiotherapists, pathologists, and clinical coordinators who cover all the aspects of a clinical trial to find out whether this next idea is going to work. And Henry Kaplan would say, it better work the first time. And so maybe it will. So this whole idea of using the immune system to treat cancer used to be a crazy idea. Nobody believed in it. And then monoclonal antibodies worked to target the tumor like Rituxan. We have many other monoclonal antibodies now that do that for other kinds of cancer. But now we have antibodies that target the immune system and take the brakes off, let it go after, do the job we want it to do. And now we have this idea I've just been talking about, vaccination by injecting take the brakes off just in the cancer area where the right immune cells are trying to do that job and help them to do that job. So I think you can see why those two guys won the Nobel Prize. They changed the world again by targeting the immune system and getting it, take the brakes off and getting it to go. And now these treatments are used all over the world in a short space of time. It completely changed the practice of oncology to add to, not replace, but add to all the other treatments that we have, the chemotherapies and radiation and the surgery. Now we have immunotherapy. So a friend of mine recently said to me, this whole idea, this whole field of immunotherapy used to be fun when it was just us and nobody believed in it. Now everybody thinks it's a good idea and everybody's doing it. So everybody's become an immunotherapy treater of cancer now, everybody in our field. And so as I said, we don't do this by ourselves. We do this with a whole cast of characters. And here's a cast of characters from our lab. Halloween, of course, we're all dressed up. And I had it on the prior slide or in this picture. And in there might be the next Nobel Prize winner. Who knows? I would actually bet on some of these people down here if I'm making a bet. So I think now that's as much as I want to tell you about it. I hope you now understand why those guys won the Nobel Prize. I think you understand about antibodies, you understand about T cells, and you understand that treatments have to be practical and affordable and available to really make a difference in the world. So I'll stop there and we can take questions. There's a microphone, and she'll come and give you the microphone so everybody can hear your question.
Okay, so if you have any questions, just raise your hand and I will give you the mic.
I actually have two questions. I'm an oncology nurse. One of them is with the immune response, are you changing? What I don't understand is when we're doing radiological studies and we've created immune response, I would think you'd have a lot of swelling around that tumor area where the immune response is happening. Are you changing the interval of when you're actually looking at the tumor response?
So, yeah, I'll repeat the question so everybody can hear it. When we do an X-ray and measure the size of the tumor, are we sure that we should do the same kind of measurements we usually do? Because maybe the immune response is now coming in, killing the cancer and filling the space that we usually see on the X-ray. I think that's what you're asking. Mm-hmm. And we know that this can happen. I didn't point it out, but on that slide of melanoma, there were some tumors that were growing and then eventually shrank. And imagine the courage of the doctors who were treating those patients, who kept treating them, even though the tumors were growing. And what turned out to be the case is exactly what you're saying. The immune cells were coming in there, and the tumors didn't look like they were shrinking, but in fact, they were being killed by those immune cells. But the X-rays didn't show it yet. Eventually, once the tumor went away and the immune cells went away, the X-rays showed the result. We are looking at the same intervals of time that we normally would be, but we're cognizant of we're aware of the fact that maybe we get fooled, and maybe we shouldn't give up too soon. Keep treating, even though it maybe looks like it's growing. It's a great question.
So, and I have a second question, if that's okay. So if a tumor is refractory, if a tumor is refractory, say they've been radiated, can you still get the immunomodulators in that radiated area to be activated?
Well, if the radiation works perfectly, there's no tumor anymore to be injecting. But if we time it right and do it before, the radiation can be in there doing damage immediately, but the damage doesn't kind of show itself for maybe days or weeks later. If we do the injections right then and there at the same time, that's what we're doing. We're talking to the immune system that's there, and we're killing some cancer cells with the radiation, spilling, let's say, the antigens, the targets, and getting the immune cells to see those targets, and that they work together that way. The timing is important to get it right. If you wait too long, of course, that tumor you radiated might be gone. You might have to go to another one and do it somewhere else. So we time it closely spaced with the radiation treatments. The radiation is, I don't know if you know this, but lymphoma really responds to radiation, but only in the place you shine the beam. And so other cancers are not so sensitive to radiation. So we're using very low doses of radiation right then and there where we're injecting the material to take advantage of the fact that lymphoma can respond so well to radiation. So that's a special case.
So a couple of questions. The initial thing you talked about, the monoclonal antibodies, and it worked for rheumatoid arthritis and some other autoimmune. Is there any work similar to that for IBD, for colitis and Crohn's?
Well, we have antibodies for colitis and Crohn's, IBD. We have Enbrel, and we have Remicade. I don't know if you know these names, but these are antibodies. These antibodies are targeting products of the immune system that are irritating the colon or the gut. And it's the products of the immune system, tumor necrosis factor, TNF, that the antibodies are taking out of the system and neutralizing those things. And they're approved by the FDA and used all the time. They don't always work, like everything doesn't always work, but we have antibodies that treat those diseases by targeting the culprit that's caused, that the immune system, overactive immune system is making. The products that they're making, we're targeting those products.
So the anti-TNFs, which are systemic, and then vedolizumab, which is more targeted, those are all in the same sort of strategy, the same approach?
Yes, you see, once the problem of making antibodies pharmaceutically were solved for the first one, people then decided, oh, this is a platform technology. We can make all antibodies now. All they have to be is specific for targeting whatever we want. But chemically, they're pretty much all the same, and we know how to make these things now. We know the genes, we know the cells, we know how to grow the cells, we know how to purify this product, and they're all the same, essentially, except for their Velcro on their tips that have specificity for the target we want. But it became, now people think this is easy to do. And there must be hundreds of companies making products out of antibodies now for treating all kinds of things. You know, macular degeneration, multiple sclerosis, autoimmune disease, infectious diseases, all kinds of things, once it became known how to make these things.
So you talked about kind of separately where you let the immune system go wild for those cancer cells, but it can cause autoimmune conditions. Is there a way to actually slow down the immune system in a targeted fashion to kind of achieve, to solve
Yes.
the autoimmune problem?
Yes, so this is the yin and yang, two sides of the coin. As I said, by taking the brakes off the immune system, we're causing autoimmune diseases. So now we have to undo the brakes sometime, put the brakes back on sometime. And so the very target that Jim Allison, the singer in the band, the very target he discovered, he used it as a target of an antibody to block it. That same target is made into a product that's used to treat autoimmune disease to put the brakes back on the immune system. So that's a really good example how the same system, you can play two sides of the coin depending on what you're trying to get to happen. I work on cancer. I want the immune system to go like crazy. But for people who work on autoimmune disease, they want to shut it down. So we can work both sides of this street. Yes?
Is there any evidence, particularly for the PD-1 inhibition, that getting it in earlier matters?
Yes, there is good evidence for that. And a lot of people are investigating what's the right time to do this. The example I gave were all the diseases that are working. Well, they're usually tested after everything else has failed. And even then, it's been shown that they can work. So what do you do next? You use it a little earlier. What do you do next? You use it as the first treatment, like I said with those lung cancer patients. So gradually it works to the front and maybe displacing other treatments that have even more side effects that are currently used. So that's the game. You start with the losers, you make them into winners, and then you get the drug approved, and then you start doing what's called extension studies to make the drug even more useful and even more effective. When the immune system is not beat up by other therapy, it's going to be more triggerable than if it really had a lot of prior exposure to bad things that hurt the immune system. So the answer is yes. Long answer, yes.
Two questions. First
Yes.
one, have you looked at using this PD-1 antibodies as like a delivery targeter? So like maybe conjugating them to antibodies and then using them to, instead of trying to modulate an immune response, instead like specifically deliver potentially like more aggressive chemotherapy drugs to the location of these tumor sites?
Absolutely. And this is an ongoing project. It's succeeded now many times already, where people are putting cargo on the antibody they want to deliver to the right place in the body. So drugs or radiation or imaging agents to get the scanning to be more specific. All of these things are being done, not necessarily against PD-1, but against many targets that might be useful for each of those purposes. Delivering drugs is already worked by using antibodies. You have to deliver them so they can come off the antibody, and there's a lot of technology around that, so they can be released when they get where they're going. But the antibody can be the delivery agent, and the cargo can be doing the thing you want it to do.
Cool. Second question, a little more tangential. You've looked at a lot of tumors. Are you seeing consistent CD47 expression in these tumors? I'm just more wondering because the chatter in the field if you also see this antibody response.
Okay, so now I'll explain your question to the rest of the audience and then follow it. So CD47 is a target on certain cells in the immune system, and it's on the cancer cells. It's one of these not don't kill me signals, it's what's called a don't eat me signal, because there are cells in the immune system that can eat the other cells. And the cancer can have it, this 47 is the name of a target which says, don't eat me. So there's an antibody, you can make antibodies against the don't eat me signal and get the cells that eat to go ahead and eat the cancer and destroy them. And there are maybe 5 different companies, or maybe more, making antibodies against this target. One of them was founded here at Stanford by Irv Weissman, who spoke at this event many months ago. And that's one of his current ideas that he's really pushing. So, and we have good results in the clinic already with antibodies against CD47. So that's one of the ongoing projects that may get approved as a drug pretty soon. So yes, is the answer.
Okay, well that's good to hear.
Yes to both of your questions.
Fantastic.
The questions and answers are always better than the presentation.
First, are they still using Zevalin, IDAC?
Okay, so I'll explain your question to the rest of the audience. So just like you can put a drug on the antibody and deliver it as the cargo, you can put radiation on the antibody and deliver that using the antibody. So this product, Zevalin, was one of those products that does that, and it got approved by the FDA. And here's another example of the way things can go wrong. It's a great drug. It really works in lymphoma. But nobody wants to touch it. The doctors don't want to touch it. The nurses don't want to touch it because they're afraid of the radiation. And economically, the wrong doctor has got to send the bill. The oncologist who has the patient, who wants to treat the patient, has to send the patient to the radiologist. And the radiologist injects this stuff and collects the bill, and then the patient goes back to the oncologist to clean up the mess. So you can imagine that oncologist is not going to be incented to prescribe that drug when there are many other drugs that compete very successfully with it. So it failed in the marketplace, even though it got approved by the FDA. I'm not sure it's still alive anymore. I'm not sure that any company is making it anymore.
My second question is, will this type of technology replace the whole study of nanotechnology with the treatment of cancer?
Replace what?
Replace the research behind nanotechnology with the treatment of cancer.
Nanotechnology is just a small stuff that you can inject or use. And a lot of these antibodies are being hooked onto the nanoparticles.
Oh, so they are.
Yeah, to make them go like cargo to go to the right place in the body.
Oh, okay.
When they wouldn't know how to go there on their own, you can stick an antibody on them and get them. And they can hold drugs, they can hold radiation, they can hold DNA, they can hold RNA. You can deliver genes that way in the nanoparticles, using the antibody as the guided missile to get it to where it has to go. So it's not going to replace it, it's going to make it work better. Did I answer your question? Maybe.
I've got a question here. I was wondering...
Where are you? Oh,
here,
yes.
yeah. If you do have the money for the custom tailored treatment, is that still possible to get? And the second part of that question, what is the primary cost driver, I guess, in that custom tailored, and would that possibly be something that could be sort of streamlined cost-wise?
So the answer to the first question is no. There's no one currently making custom antibodies for each individual patient, even though we know how to do it now, and the technology that we used 30 years ago has been replaced by better technology now to find the antibodies that would do the job. But it turns out the driver of the cost is not finding the antibody and getting it in your hands. It's making it to the specs of the FDA, putting it in the bottle and putting a label on the bottle, and quality control on a customized basis. That's the driver of cost. So for the CAR T cells that are custom made for each person, the price of the CAR T cells is about $400,000. That's just to get the drug made. Once you get the drug, you have to give it to the person, and when they get side effects, you have to put them in the hospital and take care of them. So the real cost is maybe 2 or 3 times that. But just to get the thing made, the specs of the FDA, is about $400,000. So yeah, maybe if you had $400,000, you can get someone to make an antibody for you. But there's no one right now offering that service. I know of currently one company that's trying to raise money on this idea. I can tell you the name later if you want to know it.
So you had talked about, so when you have multiple tumors in your body, or one starts kind of like with that mouse example, and then you get them elsewhere, sometimes the other tumors have different, variants, and so that you have to target them differently than just kind of that vaccine approach where you put it in once and it takes care of all the other tumors that form afterwards?
Yeah, so the way people are thinking about this problem now is that tumors are different from type to type. Stomach cancer are different from colon cancer, different from lymphoma. And they're also different in each individual, having to do with the genes in the tumor cell that are causing the cancer, or maybe happening as bystanders, as passengers in the genes of those tumor cells, mistakes in the genome. And these mistakes can make targets for the immune system. So it turns out the more mutations the tumor has, each individual person's tumor, the more mutations in the tumor, the better these treatments work. Taking the brakes off the immune system means the immune system has to have something to see in the tumor. The more targets, the more mutations, the more targets, the better the immune system has a chance of seeing it. This is one of the discriminators of who's going to respond and who not to these checkpoint antibodies. So that's currently one of the main ideas for how to figure out how it's working, why it's working, and I have some slides I could show, but that's basically the idea. Some tumors have a lot of mutations. Some of those mutations cause the cancer in the first place. Some of those mutations don't have any function, but they make a target for the immune system to see. And we take advantage of that. The nice thing about the in situ vaccination ideas, we don't try to identify all those targets. There are companies that are trying to sequence the genes, everybody's tumor, find those targets, and make a vaccine, again, on a customized basis for each person based on which targets are in his tumor or her tumor. And the nice thing about the in situ vaccine idea is we don't need to know, we don't know the targets. We let the immune system decide which targets it can go after. And sometimes it maybe work better than other times based on how many targets the tumor actually has.
So in some of those instances when it doesn't work, is it because it's not finding the targets and you would have to do a more customized thing for that person?
Well, one idea would be to deliver some targets to the tumor cell that the immune system can see. And that's another one of those good ideas that only has to do with work that we're working on. I think, yes.
In the end, you mentioned about inject the antibody into the local tumor to elicit immune response like a vaccine. But I think recently that I think MIT just had some kind of announcement. They have also using vaccine treatment, but not just locally, but can amplify the other kind of antibody treatment. Using the vaccine as a kind of amplifier.
Yes.
Could you comment on that?
Well, that's kind of what I was just talking about. There are some companies out there sequencing the genes of everybody's tumor, figuring out what targets that person has, and making a vaccine just from those targets for just that person. And using a vaccine not into the tumor, but in any place in the body to trigger the immune cell against that target that you already discovered and know, but just for that one person. And that's another customized approach you have to wait, it's expensive, and the whole thing about putting it in the bottle and putting a label in the bottle and the FDA, it's not going to be cheap. We don't even know if it works yet. So it's still in the testing phase to see if it can work. I think probably we've exhausted questions. No, there are more questions. Okay.
When you first talked about Rituximab, you showed that there are all those other things that fortuitously it seems to work on. Is there any more work being done on that, or is Rituximab sort of just there until something else replaces it?
Well, there's a lot of work on that going on. I talked about inflammatory bowel disease, other autoimmune diseases for which we have different antibodies, not rituximab, that go after what makes those diseases happen. And as you can imagine, rituximab was approved in 1997 by the FDA. 22 years have gone by. It's currently off patent, and there are a lot of copycats being made to either improve on it or be better or be copies, me too, me better. And there's a huge industry out there. That $5 billion market is not to be sneezed at. So it encourages a lot of people to go after that.
Well, in my case, I have IgG4 related disease, which is one of those rare conditions. But as far as I know, Rituximab is the only thing that seems to work on it. I don't know of any research being done on it.
I don't know of any specific research being done on that particular disease, but if it's working, keep doing it.
Oh, yeah.